Great. So my clock says 10 o'clock or on the hour. So let's go ahead and get started. Welcome everyone to the B Short course for community scientists. I'm Denise Ellsworth with the Ohio State University Department of Entomology and Extension. So welcome. If you're returning to this webinar series, welcome back. If you're new, we're glad to have you. All of our sessions are recorded and posted. We have a web page that's up on the screen there, the u.osu.edu/B course. All the recordings are on there, and they're also on our YouTube channel, so you can access those recordings. If you've just signed up and you're just joining us, there's no particular order that you need to do those sessions, so feel free to just run through it depending on what you're interested in and access those webinars. So we just have a couple to go. Sam Droege is here with us this morning. I'll introduce Sam in just a minute. Next month is going to be Dr. Mary Gardner, who's with the OSU Department of Entomology, talking about the importance of community science in the world of entomology. She's got some really nice examples and links to share with us. And then our last session, we had a change in speaker, but we're still having an expert, Matthew Shepard from the Xerces Society. We're excited to have him, Director of Communication and Outreach for Xerces, and he's going to be talking about the way that community scientists add to advocacy and change on the ground for pollinators. So excited to have Matthew stepping in for that last session. And we are working on a bumblebee course for next year. So if you've enjoyed this monthly series, we're going to do a similar one. You'll all find out about that as it comes together in the new year. A little bit about our program. So we are doing these monthly webinars. As I said, they're recorded and posted on our YouTube channel. We are cooperating with the Chadwick Arboretum, which is our arboretum here at Ohio State University on the Columbus campus, and also the U.S. National Native Bee Monitoring Network, which is a consortium of bee researchers from across North America who are coming together to share resources and put the brightest minds in bee science together to help with native bee research. And so this series is really aimed at community scientists to help pull all of you together so we can build our skills and add to that body of researchers out there doing important work to save native bees. We have some funders that I want to thank. We get funding from the USDA, a NIFA grant, an integrated pest management grant that specifically funds some of our outreach in pollinator health. So appreciate that funding, and also the Davie Expert Tree Company that's helped with this webinar series. I want to thank and welcome Marcia Carson, who is a facilitator in this program, helps me here with a lot of our Zoom details. Marcia's been here every month and lots of other webinars that we do as well. Marcia, thank you so much. If I crash, I know I have a great backup in Marcia. She is a volunteer pollinator specialist here in Ohio and also an advocate with, or an ambassador with the Xerces Society. So Marcia, thanks so much. So if you've been here before, you know that we use that chat box for city and state or city and country just to see where folks are coming from. As I said, today we're going to go ahead and keep the chat box open at Sam's request. So he'd like some input and some suggestions for resources that you all would like to share. So you want to find that chat box. And then we use the Q&A box for questions. We use the upvote. So as you go into the to the Q&A box, if you see a question that you'd like answered, be sure to upvote on that question. It'll help the question rise to the top. What we'll do once I introduce Sam, he'll go through his content. He's going to take us on a nice tour of some of the resources there in his lab. And as he gets to about the hour point, we'll switch over to Q&A. I know that your time is important. You may have other meetings or obligations this morning. And if you need to hop off, just know that that Q&A portion will be recorded and you can go back to that, hopefully if the tech gods are smiling later this afternoon. So I'd like to introduce Sam Droege, who's really one of my bee heroes. Probably lots of you are familiar with Sam and the important work he does with the USGS Bee Inventory and Monitoring Lab. I first met Sam in person a few years ago. So not only is he an amazing bee researcher and resource, but he's got a really strong background in bird conservation and also phenology. So we first met in Cleveland at a phenology conference where we were both talking about some of the phenology work that we do. But we had taken a break for the session. We were out at the Cleveland Botanical Garden, they had one ironweed plant in front of their entrance, and there was the specialist bee right there. And Sam points it out, and he said, "Do you know this bee?" So I always think of Sam when I see that bee. Sam's one of these amazing people who shares so much of his knowledge in a lot of different ways. He has an amazing Flickr page with an incredible source of bee images. Let me forward to this image that shows some of that photography that Sam has offered. Those images are up there as Creative Commons, and so you can use them in your programs and other, in your writings and other needs. Not everybody shares as widely. I just love that they're all licensed community commons. He also has a lot of manuals. This Bees of Maryland Field Guide is out there. He's shared that resource with us here in Ohio, and we've created a manual for Ohio. And then you've all got the link to the Handy Bee Manual. And if you scroll back up way to the top on the chat box, and we'll put those links in again, I have live links to all of those great resources. I also have those on our Bee Course webpage under the resources, so you can go back in and check out some of those links later. So at this time, I'm going to turn the program over to Sam, who's going to walk us through some of the tips and tricks from his great resource of bee knowledge. So Sam, thanks. It's so nice to see you, and great to have you here this morning.
Thanks, Denise. And I have to throw some compliments back, because you guys have really done a great job in Ohio in activating a group of people, training them, and developing information about the bees of Ohio. And very few states have done it to that level. And that's where our needs are, is, how can we conserve something we know nothing about? So a lot of this is getting boots on the ground, and we'll show you today some of our tricks that we've learned over the past 20 years now about, I guess, maybe some mundane things that are useful. And so, yes, I ask that the chat stay open, because we have certain approaches that we find useful for how we run our workflow, but other people have equally valid and equally interesting, and maybe stuff that we'll want to grab and once we learn about it. So please add I'm like, I use this, here's a picture of that, here's a link to something. You can get these kinds of tweezers from these places into the chat box, and maybe Denise will compile some of that information too at the end. So another thing, I just saw a flash through on the chat about our photography technique setup. So if we have time, I can move the laptop here over to our incredibly complex photography studio, which is basically a beer cooler. And talk about how that's done pretty quickly. But because we have a lot going on, I'm going to jump right in. We're going to start with, and also I'll need some feedback from Denise and Marcia. I have to tuck my pants in, of course. Boy, this is awkward in the new video land. But about how I'm doing, I'm normally not a live caster of showing you things on video. So if you want, maybe I'll put this a little bit higher. You can say, bring that closer, hold it still, any of those things. My notion is that maybe I'll move too fast for video feed sometimes. So just give me some feedback, and people in the chat box can give feedback like, oh, he's going too fast. So we'll see. So I'm going to start first with collecting in the field with nets. That's the tradition. I'm going to show you my favorite net here, which is was done by Rose Entomology, out of business, but now run through BioQuip. So for those of you who don't know BioQuip, so someone should be typing in the BioQuip link in the chat box. That's the main US, and I would say Canada probably, supplier of entomological equipment. A lot of what I'll talk about, that's more the real hardware would be gotten through there. This model, I think, is out, but I really like it because it's got two extensions. There's another extension that can go out. It's got a very stiff rim. So a lot of the type of nets that people use, which I have examples of over here, are going to have flexible rims. And which do have, so this is an 18-inch. This is at the largest size, really, that's practical. I like it because it gives me a bigger area, but it's more tiring to swing. It catches more wind volume. So a lot of people are using 12 or 15-inch, which are sort of the standard size BioQuip models. This is the BioQuip standard net that you would purchase. It's got the red twist, if I can show this right with my finger there, wrist twist top, and there's a strap that goes around. You can take this apart and put it in your luggage to some extent, and it's a very flexible rim. That's pretty good for lots of circumstances. It's great for slapping on the ground because it does some conforming to irregularities, but I actually prefer, this is, I think, aircraft aluminum rim, so sounds expensive and it's more expensive, but because it's absolutely stiff, and I have broken the tips off, right? So right here is where it goes into the joint. I did break eventually, and I'm using this constantly at some point, but it will crash through vegetation. So even relatively stiff woody plants, you can plow through, and we won't go into a lot of detail here. There's videos that we have about doing that, but you usually center the bee on the middle of the net and you plow through the vegetation to pick it up, unless it's got stickers or thorns on it. And then there's different approaches, which I'm going to skip over for now. We can return to that. But this stiff rim is really useful. The extra extension sometimes is very useful on bees that are always just a little out of reach in a couple different ways. It's a little heavier. There is not this exact model, but there is a similar one with an extension on this that has the stiff rim that would be, I believe, still listed as rose entomology. Another type of net that's useful and used a lot is this one, which our long-term volunteer Jane Whitaker just donated to the lab because she's no longer in the field. And this is a collapsible net. So I'm going to uncollapse it in a second, but you can see it actually comes. This is also available on BioQuip, and you can see that this can go right into a backpack, and you have a variety of different kinds of extensions. So if you are working trees or something very tall, usually the cheapest and easiest way is to have this kind of net set up with extensions to whatever level you need. At some point, it becomes very wobbly, but your options are limited if you want to reach high. These are even sometimes if you have a little day pack, it actually can fit in there too. It's very handy. So this just folds up and flips back out. This is a good example of a net that Jane particularly likes because it can handle thorns a little bit better. So what you have is a canvas upper part, and the lower part is the traditional aerial net. So this doesn't snag as much nor tear as much as the traditional net bag, such as a lot of our nets are filthy from use, but such as these are really prone to getting snagged. And after a while, you get good at both spotting the snags and backing out of them, but still, particularly when you're beginning, this combo bag of canvas or cloth or thick cloth at the top and a net bag at the bottom, useful. So I'm going to stop on the net front. We can return, if time and interest, to talk about approaches to catching bees. We have some of that on video already. So once you're in the field, what people do is, tradition would be you have a kill jar. It's charged with cyanide. It's charged with ethyl acetate or a couple other kinds of combinations of things. We started out that way too, but now we actually I haven't used kill jars in quite a number of years. We find that putting everything into liquid, soapy water essentially, is the way to go. Our favorite, for a couple reasons. So our favorite container is a simple centrifuge tube, 100 millimeter, I think that, or 100 milliliter, or 50 is what that one is. You can see by the size of my hand. These are super common. I would suggest going online and looking for used ones. This might be a good group purchase through Denise's group, or someone should do group purchases. And then you can divide these up because they're cheap when you find them in gross, and they're often in surplus. And if you're associated with any kind of lab, these things, like we have almost an infinite number of them. Actually, I'm not giving them away, so I don't have an infinite number, but they show up in terms of lab surplus all the time because people buy them by the gross and then end up not centrifuging as much as they thought. So you fill that with
management team. You should be using Monday.com. If you
are we good? Okay.
Sorry, we're good. Yeah, I was going to your YouTube channel and I got the commercial. I was trying to get your link. Sorry about that.
Oh, okay, no problem. Anyway, it's just, I fill it up about that much with water. This is really, there's so many different ways to run this. And then I add some kind of soap. So soap in this circumstance doesn't matter. You're just getting rid of the surface tension. If you don't have soap in it, your bees, first of all, don't expire. That's my phone, just ignore that. Don't expire right away. The soap is actually a killing agent, and they don't go into the water very well and sink and allow you more room for collecting because the surface tension is there. So the procedure in general would be, I've got a net. I am going to have this net, and I'm going to keep collecting. So normally you would catch a bee, take a bee out. If you have a killing jar, it expires, then you can do another one. But in this circumstance, I'm going to continuously be catching bees. I'm going to snap them down to the bottom, grab that net, all the while looking for more bees. I thought I said I wasn't going to talk about this. And then I'm going to catch more, and I'll have a net full of bees and sometimes other insects down at the bottom. I can snap that down like that to keep the stinging things down there. You can wear a glove if you're nervous about that. Then I'm going to hold it against my chest. I'm going to take the vial, which, because of that tip, slips right into my pocket super easily. I take it out, I unscrew it, I hold the cap next to my fingers, I bring it into the net, and then I let the bees wander out a little at a time. I make my selection for what I want. I tap it in and I rub it against the top to pull them off because they'll take their mandibles and grab onto them. And I can clean the net out in one run. So I don't have to be putting the net down, picking it up, taking my killing jar out, putting it down, because I can pull as many bees as I want into this jar. So they might try to climb out a little bit, but a simple tap puts them back. But in a killing jar, what happens is you get a chance to take one bee in, and then when you try and get the second bee, the first bee leaves. So this is the way to go, but it's going to require, as we'll talk about, dealing with wet bees. In general, always process your bees as quickly as possible if you want the best look. So,
Sam, there was a question already about why would a citizen scientist or community scientist kill bees? And I know you were going to talk about that. I don't know if you want to address it now or a little bit later.
I wasn't going, yeah. There's so many things to talk about. The best thing is maybe, I don't know if you have an email list, I can send a whole long list of discussions of lethal versus non-lethal and why you would do that. So, but to parse it down to bare bones. So it's an individual decision, and there are some things that can be done that are non-lethal, mostly with things like bumblebees and photography. But in terms of what I would call the higher value things, we have to look at them under the microscope to tell what they are. Like I can't, even if I've caught them in a net, I have no idea what they are because ID is so difficult. You can see, and I believe Denise has linked to the top of the page, a series of YouTube videos, I don't know, we might be up 17, 18 videos just on identifying North American bees to genus, let alone to species. So we'll probably have 100 hour-long videos on bee identification because it's so tricky. So I encourage people to look at that. The lethality issue, there's a whole other set of things to consider, both on the pro and the con side of things. But in a information conservation point of view, we have to end up, at least in some of the kinds of investigations, looking at what amounts to dead bees. So that's the small discussion of a much larger topic. So I'm going to move on. We can, bring this topic back up, but I don't want Denise to cut me off from all these good contexts. So we now have these so bees in a centrifuge tube, and we're going to process them in a bit. But I'm going to show you one of the other main capture techniques that are used quite a bit. And essentially, oh, let me back up. So this is a new gizmo innovation, which is a very simple thing. Is that 4-inch? I think that's a 4-inch tube. I can fit my hand in there. I've got a binder clip and I've got a Ziploc bag. So, and this can make a good non-lethal approach to some extent too. So what I'm going to do is do just what I was talking about. I've got a net full of bees. They're at the bottom. Instead of picking them all out and putting them directly into a soapy container, I'm going to have, let me set this up. Let my pretend bees go. And I'm going to take the tube. So this, this could be a rolled up piece of cardboard. It could be anything like this. This is just happened to be something we had around that I didn't have to buy. And you take your Ziploc bag, okay, very clumsily and inelegantly take your binder clip, and you just take the binder clip, and again, trying to do this on national YouTube TV and messing up. So there it is. It just tucks it in. I've got it like this. Okay, I would have it on the ground or on top of the car or on a table. I've caught a bunch of bees. Okay, now what I want to do is get the bees into this Ziploc bag while they're alive. And so what I'm going to do is I'm going to put this on its side. I've got the bag here. I'm going to let's see if I can do this. I'm going to invert it, right? So I've not been stung doing this, but you can get nervous when that bag is full of bumblebees, which is part of our standard bumblebee survey technique for non-lethal bumblebee sampling. And I'm not showing you the entire fast way to do this, but I'm going to, so you might have a glove on, is all I'm saying. You're going to stick your hand and the bag in there. Now I'm inside the plastic bag. I've got the bag, it's still inverted, and I'm going to pull it off my hand by pulling it backwards. That is releasing all the bees and whatever else is in there.
And go up just a little bit. We're cutting. Yeah, there you go. Thank you.
This bag. So now there's all kinds of stuff flying around. I'm going to wave this madly around to get everything to the bottom of the baggie, and I'm just going to pull that baggie off, and I'm going to just hold the top shut, get rid of the binder clip, and with a little futzing around, now I've got them zip-locked in there. If I were really clever, I'd have a note and pencil inside already. Now I can very much more conveniently than in a net, look at what I've caught. So for a bumblebee survey, this is our standard protocol now. You simply look at the bumblebees in there and divine what the species are, and then you let them go.
Sam, I want to mention the zip net instructions that you have in the handy manual, because I modified my student nets as zip nets. And so when I'm out in the field with people, they have the Ziploc bag at the bottom of the net, and they're same thing that you were talking, except not quite the tube and inversion. The bees are right there in that bag. It's really great for kids or people who are a little nervous about catching bees. Yeah.
And so, so if you have a modification, send it to me so I can put in the next version of the Handy Bee manual, which we're working on, or you don't.
I'd use what you recommended. Yeah.
Okay. So here, so this then I can now take this off as I could with the modified net, which a lot of people don't bother to do that because that's a lot of extra work. And I'll have a tag inside, paper and pencil. I can put it in the freezer, and now I can look at those samples later. So we're using freezers as our main storage container. We're actually most of the time not even using alcohol or propylene glycol for any kind of storage. It's just freezer work. So, but this also could have been filled with some alcohol or propylene glycol to save a sample, or you can just use it to visually look at what you have and release them. You could take pictures, but I can tell you that this often will fog up, and it's just not a great photography thing. You can do things like have a pooter, which is look it up. It allows you to suck insects out of small areas and into a vial. We don't actually use them, but you can do that. Then you can pull out which insect you want by snaking that in there and moving it to a vial. But it gives you several options, including a lot of non-lethal stuff. We use it for bumblebee surveys, particularly in Bombus affinis areas where, we can't make a mistake of killing a Bombus affinis by accident. It's surprisingly difficult, even in a net, to tell whether you have Bombus affinis or not. So that's netting things. I want to talk now about bowls. So there's lots of different ways to do this. Basically, you have a white, a white, a yellow, a blue. Those three color combinations maximize the spectrum of attraction for bees. We put those out in different kinds of combinations. They're filled with soapy water. We always tell people to use and the government is not championing this commercial product, Blue Dawn, because it's so available and has not a citrus scent. It has a duck scent, apparently. But the reason is citrus oils are repellents for insects. And we have shown through some of our student products that using a citrus-based detergent decreases your catch by a lot. We have several other bowl innovations, but our problem right now is this is the only kind of bowl you usually can get in those sizes in general. Those are 3.25 ounce deli cups, usually made by Solo, but other manufacturers. Translucent paint does not like to stick to that. This is our favorite primer there by Rust-Oleum, very available. Almost every hardware store covers it. We've tried a ton of others. This works the best. Again, we're not advocates for Rust-Oleum. And we use from Guara Paints, which is in the Handy Bee manual, a fluorescent yellow and a fluorescent blue pigment. We mix them into the primer directly and usually strain them out because this pigment is clumpy if we're going to put it in a paint gun, but you can just paint them. There's some more nuance there. We have unpublished studies that show that if you paint the inside of these bowls, that some bees will climb out because they now get a grip. But if you have painted the outside of the bowls that color, the inside is so smooth that no bees get out. So you'll hear about that. I encourage you through the links or emailing me that you join our bee monitoring listserv if you're into this sort of thing, because we'll make announcements about these techniques. And there's chatter about survey techniques and IDs and the mundane of how-tos. So
Sam, there's a little bit of interference in the background. Do you have a video going, or maybe it's just a conversation?
I have people going.
Oh,
so,
gotcha. There's work happening.
Okay, so I told them, I was going to fire them if they talked.
Thank you.
That's how we roll here. So when you have these bowls, traditionally, in the Handy manual, I think largely talks about that you would dump the bowls into a brine shrimp net. Brine shrimp has smaller holes than regular ones, and then you have to extract them and put them in another bag. Shortcutting several things, you can An easier one is to use what are paint filters. And these are used by automotive paint people. You can buy tons of them in bulk. And there's a tiny screen at the bottom. You want the largest mesh size, which I think is the smaller number on these, because it's so fine that often it will be slow. You can dump your bowls into these. You can write on here in pencil, but this paper does deteriorate. So you want another tag, which we'll talk about in a second. And then you just fold this up. And if you're nearby, you simply fold it over on top, staple it, and put it into a container, just an open container, and then take everything and put it in the freezer, not into any more liquid or a plastic bag. And the bees will have partially dried out and will be a very nice specimen in that way. If you get things out of liquid and dry and process them within 24 hours, your specimens will be primo. Any longer than that, the hairs become very matted no matter what. I'm not sure why, something about the hairs there. But our latest thing is that we use, where's an empty bag? Oh, here we go. So one of our volunteers is very good at the sewing machine. So we take mosquito netting, and I mean, this is something anyone who knows how to do sewing, this is like sewing 101, is you make a bag with a drawstring on it. So in the field, you take a flower pot, or you take whatever kind of container, like here you can see this container, which had some mysterious commercial product in it. And then you drill holes in the bottom, you dump, so the drawstring is over the top, you just dump into this your specimens. Then at the end, you pull it and you tie off the bag and you wrap it up. And but before you do that, you add a specimen tag that you've written out in pencil, and we can send these to you. But one of our problems when we work with people, particularly if we're asking them to do things, is they send us mysterious notes with their data on paper that we swear they wrote it on toilet paper and is now just pulp. So tough paper, a standard form by whatever your project is, and you want all that information like latitude and longitude, lots of redundancy, because I can tell you that all of your participants will mess this up in so many ways. So you want to eliminate that by giving them the forms, not telling them what to write. And then that form gets cut out, added to the bag, and then brought back to the laboratory. We then, here's some specimens. We then do something like this. I'll just use one bag as an example. We will have a bag now, and we throw this into the freezer immediately, okay, when we come back to the lab. We then pull out the tag, and we add the tag now to a Petri dish. That is not an ordinary Petri dish. It is a Petri dish with a set of colored rhinestones glued to the bottom. And those colored rhinestones are matching a set of colored rhinestones in this bag at this point. And we then take a series of these bags, throw them into the freezer until we have a bunch. I take them home, I throw them in the laundry machine, I run them through a delicate cycle, I bring them back. We have a little mini dryer, like a desktop apartment dryer. And then we throw them in the dryer somewhere between 10 minutes, 7 minutes to 15 minutes, depending on size, amount, and number of bags. And we have beautiful specimens that are bouncing around inside the bags, fluffing their lovely bodies. And we don't have, like we do, which I'll show you in a second, if you're doing individual specimens, problems with people shortcutting the drying process. So the traditional way would be canning jar, screen on top, usually fiberglass screen, cheap from the hardware store. And you can glue it, or you can just simply clamp it down there. Specimens go in, and then you use a hair dryer, which you know what that looks like, or a big blower, and dry it by hand while shaking the jar so the specimens are bouncing across the screen. You want them to have their hair tousled. You don't want a bunch of matted hair on the ground. So you want active processing. These are slightly better. So PVC pipe fits together, screen on the bottom, glued with pipe glue, which will glue the fiberglass stuff on too. You put them together, specimens are in there, you blow through there. There's a lot of more airflow and bouncing around. Be careful, you can do that too much and pull off a lot of tarsal claws.
Sam, let me just jump in. I wanted to mention to everyone that I will record and post the chat. I'll copy and paste the chat onto our recording page. So if folks are trying to figure out how to copy it, I can't enable a setting that lets you copy the chat, but I will post that for you.
We do that for, like, if we have a special batch, we want to look at the bees right away now, but largely we're going to the net bag thing because it's so much more convenient. And each of these steps can be and should be put into the freezer. So afterwards, we end up with this is empty. We end up with a Petri dish. Usually we tape it so it doesn't open up after it gets jostled, with the label reconnected with the dried specimens. We put them in the freezer so they don't completely dry out. And then when someone comes into the lab, they know they can pick up this set of specimens and process them, and because they're in the freezer, they're still fresh enough to pin, and we do a lot of gluing. So I think I'm going to now go on to the process of pinning. I'll say that we keep track of each collection event and give it a separate number. So every time I or anyone else goes out, does a set of bowl traps at a different location, time and place, it gets its own number. You'll find that ultimately that's very useful to do rather than simply numbering each individual bee and tracking that somewhere else. So we have two numbers, a bee number, unique, a collection event number, also unique, a useful addition. So pinning wise, let me say that we have for years now used this brand of pins. Government does not advocate this brand of pins as a government, but Sam does, because they are super cheap home tools, way cheaper than, as far as I can tell, all the other brands. So that's useful. We use little holders for pins, but what we do is, first of all, let me show you why we use pencil. This is our classic that's in the bathroom. This is someone sent us in a whole bunch of specimens. They were in alcohol and they had filled it out in pen. There were words in different lovely different colors in their Sharpie or whatever it was. They're all gone. And people will write on the outside of labels, no Sharpies allowed. Sharpies do not really perform well with plastic and with alcohol in particular, and sometimes not even with propylene glycol. It's a all pencil thing. If you want, you can write something on the outside of the bag, but there has to be a pencil label inside. So we use cross-linked ethafoam, which in any large city, you'll have a foam manufacturing plant and they'll do whatever foam you want. Cross-linked ethafoam, we use dark ones so it doesn't show how dirty everything is. And sometimes they have overruns and they'll just recut their runs, because when they're manufacturing, apparently they're doing everything like in 3-foot thick intervals. They'll recut it to what you want. 3/8 inch is your diameter. And then we glue that to lots of things. So the first thing I'm going to show is that for some reason, these signs, which were along highways illegally, ended up in our lab, and then we glue them to the back. So with cross-linked ethafoam, unlike styrofoam, you can use white glues, but you have to weight them down. Mostly we are using hot melt glue guns. Get a good one because they will gum up if you don't. And we are using construction grade glue, not the crafty stuff, which is basically just melted plastic and will eventually crack off. Anyway, so this is one of our worksheets. We use parchment paper, which we use over and over again on here because glue doesn't stick to it. And I can literally move the specimens with my hand wherever I want. I don't have time to show you our sorting, but we'll line up the specimens out of that bag in a line down here. And then with my finger, some people use tweezers, but I move my fingers. One side are bees, others are bees or things I think could be bees. So it's always important to be conservative. And other things go the other way are not bees. The not bees go away in our lab, and then the bees get processed to pinning or to gluing. If we're gluing, this is our gluing sheet 2, and you can see things on here that say, do not pin through the sheet. So, and that's just one of these, it's a tip throw, right? So that if you start pinning through this, then it catches, those little pinholes catch on the little tiny legs, and so it ruins them. You pin to the side if you need to, and then things are glued on this sheet, and we have videos on this, but basically we are using a glue similar to this, a tacky glue. Is that upside down? Yeah, tacky glue. One of the ones, look for archival quality. You can use white glues, particularly again, glues that are archival or PCAs, but they often don't completely dry crystal clear. These dry crystal clear, which is helpful when you're trying to see through the glue if there was a glue got smeared in a place you didn't want to see it. So this type is what we're using. We will open it up. We'll run a line of glue down our thumb or a forefinger. That sounds bad. A line of glue. And then we will take a pin, pretend this is a pin, and we will touch the pin to the glue line at the level we want the specimen. We will go back to our sheet, touch the bee on its side or underneath, never on top, and then we set it aside in lines to dry on that sheet. And then in about 15 minutes to whatever later, we can pick them up and move them to boxes. When we do that, here's what things look like. First, let me talk about boxes. So we use pizza boxes, and we on our third pallet load of these things. It turns out that if you use the standard BioQuip box, which a lot of people use, it's a lot taller. Okay, so it takes a lot more shelf space. It also is equally prone to getting insects, which climb through that, which we'll talk about in a second. And it is expensive. This is a lot less expensive. You can actually, if you're not doing a lot, just bum them off of pizza people or eat pizza carefully. And then you remove the top because you don't want that attached top because it gets in the way when you start spreading things out. So you cut it off, you get rid of one of the tabs, and then you have foam, hot melt glued to the interior with areas to either side that the lids of the edges of this will fit in. You then have this situation. Okay, here's a collection event label, and here's a series of specimens that are now in some combination of pinned and glued next to it. We do rows of 10, so it's easy to count, right? So because we're going to assign each of these labels with a collection event number, and then we're going to assign numbers to each of these, but we have to count them. So another tip, rows of 10 makes everyone's life a lot easier versus, big jumbles of specimens. These are our labels now printed out here. And again, this then becomes part of the workflow. This box will go through our system together. Someone, because we have lots of volunteers, can come in and they can associate the group of labels to the specimen, to the collection event number, cut them out with very small craft scissors. So these kinds of scissors are super handy. And so they'll cut them out and label them. When we're cutting out specimens, we will cut a line of labels. Let me show that better. So much to cover here. Here's a set of labels, as often are, however you generate them, it can look something like that. We cut them into strips. We put a strip down in another box. We move the collection event label with it, so that helps track things. Then we put the specimens in on the label with the head towards the matrix, all in the same row, but it's not cut. Then we'll either, on a row at a time or 2 rows at a time, take those little scissors, dive into the foam and cut them in place. One less move that we have to do, and it's fast.
And Sam, just to clarify, you're doing lots and lots of samples, right? You're processing hundreds of bees.
Like I'm saying, this is our workflow, and that's why the chat thing is useful. Like we're showing you where we, our lab motto is, how do we increase our bees per minute? So lots of these little things make less work for us in the aggregate, because we're doing thousands and thousands of specimens. And it may be that at the level of an individual, it's not so important. But on the other hand, most of this could be done at that level.
Before you move on, there were a couple questions related to processing the specimens. How long can the bees stay in soapy water, and how long in the freezer before you need to take them out and process them?
So in the freezer, it's indefinite. In soapy water outside, or in your vial, surprisingly long periods of time. Jane, who is our West Virginia volunteer, would put cottage cheese bowls that she painted out for 2 weeks in the summer, come back, and yes, they smelled, but they actually processed pretty well. So it's amazingly long time. They will, essentially they're going to start decomposing, but the chitinous integument is actually pretty tough. So, but in general, it's good to, if you have the time, to process them within 24 hours or less, because you get good looking the best looking specimens that way. You don't, if you are precise about how you wash and dry things, we can talk about propylene glycol because it's a special case if people are doing that, but you need to be good at washing and drying, or your specimens look terrible. You never want to go, here's a bunch of specimens in soapy water, sticking pins in them and putting them as your final product, because they will look terrible. The hair will be matted. It'll be discolored because it's matted. The wings will inevitably become glued to the side of the body, and it just makes identification terrible and aesthetically, like, who wants to look at that? So does that answer?
Yes, it does. But can you do your quick elevator version of how many bees there are per acre? Because there are a couple of questions about, like, why are we killing native bees, and are there enough bees to be doing this?
Yeah, so we have we have lots of information, and I can send the Excel spreadsheet that shows how we approach this. But our back of the envelope calculation is the average acre in the mid-Atlantic states, so this would be appropriate for Ohio, is that an average acre is producing on that acre about 26,000 bees. We really intersect with them relatively rarely. So even going out and trying to capture them is very inefficient compared to the numbers that are out there. A lot of these bees are out for only small periods of time to gather their pollen, and they're back in the nest. They're hard to find, hard to see. They fly different distances. So it's very difficult to almost impossible, really, to collect out a population of bees. So when we're sampling and we go like, oh my God, I got 1,000 bees on my project, like, that's a drop in the bucket. And there's also a whole series. So we're not talking about a moral, I am morally opposed to killing animals. We're talking about a biological impact in later populations. Like, are we harming these populations? And the answer is no, because we can't catch enough bees to have an impact on the next generations. When we catch bees, a chunk of them are males, so males are not monogamous. So there's always pretty much enough males to get the mating done. And actually, an unmated female will just make more males because a male is an unfertilized egg. Two, when we're catching females, because of the way the average solitary bee is working, which is almost all the bees, is she makes a series of nest cells, lays eggs into those nests, and seals it off, and then she has no more interaction. So on average, she would have produced about half of her next year's offspring too. So biologically, again, not a moral inquiry here. Biologically, we really have no impact. From a statistical, scientific point of view, because there's so many bees out there and we want to understand what they're doing, how they're doing, what the species are, you want to catch lots of bees. And to understand what the species are, that right now means they have to be killed in some way. So the lethality of a lot of these techniques is sad, I would say, and I feel that, but it's, if your goals are really to help these bees, you have to know what's out there. And right now we're stuck with the lethal things. How's that?
It's good. Thank you.
Okay. So continuing on with processing. So once they're labeled, then we, I won't show you the process. They're labeled. We'll have people in the lab who may not know anything about bees morpho sort them. So that helps with the identification ultimately, because all the big bees will be in one place and you have fewer moves that the identifier, who is the weak link in almost all these systems. It's easy to collect bees, really, particularly with bowl traps, but someone has to still struggle through the identification. So anything that can be used to shortcut that is good. So a lot of times we collect the entire collection of all the bees from a project, and then the interns and the students and the technicians will morpho sort those, and then they're given to either Claire or myself to ID, and that's a big boost in our productivity. So ultimately, what you end up is with a box that might look something like this. So what you're seeing are, maybe not well, are specimens, and specimens where the label is vertical, those are females. So we don't, again, we're busy, so anything that we can do, so having to separate out males and females or write their names out is a waste of our time. Well, it's time consuming. And then horizontal are males. And so, and then in front, reading like a book, right to left, top to bottom, is a label that identifies what the species is. So it gives it a name, and then whoever, and it's not under the specimen. So traditionally you would put the det label, it's called, under the specimen. And, but it's hard if you're doing data entry to read that. In the old days, no one did data entry. You just put them into unit trays. So here you have the det label, and then the person who's doing data entry, and you can see there's a QR code or a matrix code on each label. Maybe it's hard to see, but there is. And so probably not something most people are doing, but if you're part of a larger group, you would be probably wanting to do this, is you then have a simple scanner that we buy used, and you can scan those codes right in so you don't make mistakes typing in 6-digit numbers, which is inevitable. And then we have a whole process of, I can show you our stacking routines and things like that for data entry and double checking. Part of some of these processes is going back or during the labeling process and saying, you know, that specimen, that needs to go to the Smithsonian. That specimen, it needs to go into our collection. That specimen needs to go to so-and-so for more ID or for some kind of confirmation or a special project. So you can use different colored pins with pieces of paper on them to stick back into your collections that might be, oh, take a picture of this, or something's going to be done. So we use these, and also, you know, little stick pins of different kind to mark specimens in boxes so that we don't overlook them if they're special for some reason. Just, we were talking about those jewels. We keep our jewels in little containers so that they can be refound very quickly. We have microscope stuff. We buy microscopes used. There's not one microscope in the lab that wasn't purchased, used, or given to us from some other group. They're very sturdy, so used microscopes are a good value. And so if you go online to use scientific equipment dealers, they have to deal with reputational things. So we've never had a bad purchase. Right now we're fine because we get these things for free. My suggestion is to look for microscopes if you're going that route, which I always encourage people to do online. There are often very expensive lights, but we use different combinations. This is the Lawrence Packer Lab approach, which was prior to LEDs, and I think they still use them. This is just a gigantic compact fluorescent of like 150 equivalent watt bulb, and on a standalone light, extra weight so it doesn't topple over. Also, you have to make sure the bell is ventilated, because a compact fluorescent of this size will produce a lot of heat at the base. It's clunky though, and you often have to put in shading. What most people, I think, are going to, if they have any sense, is this. Again, there's other ways, but IKEA has these kinds of student lamps of different kinds. Some have 2 on them, and these were $10, right? And so this is basically all we use now. I can gooseneck this to wherever I want in terms of the angles. Under microscope, we use 2 usually because that gives better light. A long time ago, I realized that the flashlights, the LED flashlights that were coming out, were actually produced really good light. So this, you can see here, is a very cobbled together system. I have a botany far reach thing, so it's right next to my desk, so I can pivot this back and forth. So this is a weighted stand that was used for botany thing. It had a different scope on it, which I've jammed together with pieces of foam. Here is a Gerber REI, 20 years old now, light that I wired together in some cob job way with one of those little wall cube converters. So you can look at the batteries, figure out the voltage, find a converter somewhere that matches that, and you can make it into an electric light. The IKEA lights replace those, but this is still a totally good light for me. And it also becomes portable, right? So there's another odd tip. Now I've tipped my whole laptop over.
Sam, you hardly take a break, so a breath, that is, so I can't even get in. I was going to give you a 5-minute warning. Now I have to give you a 60-second warning. So a lot of the questions in the chat box we've kind of answered as we went. I'll keep looking at those questions. Okay. But some of the concerns from some of the participants was, these are pretty advanced things. Are there simple things? How do I know? How do I bridge this? How do I get started?
So the basics are a net and some vial with soapy water, I would say, as a starter thing. Then to wash and dry your specimens is just the canning jar with the screen on top and a blow dryer. So that'll get you need to buy some pins, and then you can put them in all kinds of boxes. We suggest doing the ethafoam thing, or you can just, again, if you're just starting out and you have some money, just buy the BioQuip boxes. Be aware though that one, mice love specimens. So you should put them into a mouse-free environment. You can also, there's dermestid beetles and a couple other things, but mostly dermestid beetles will attack these dried specimens inevitably. And you can, we allow a certain low level amount of pests in our lab because we have so many open boxes, but we rotate everything through a freezer. So 3 days in a freezer is enough usually to kill all the eggs and larvae of dermestids, and it keeps our populations low. You can do the same or freeze them once and then put them into gigantic Ziploc bags. You have to be aware though that if the specimens really aren't dried out, then you could end up with mold in there. A general thing is wherever the specimens are should be in an air-conditioned space in Ohio. Well, actually anywhere where you have significant amounts of humidity. So I made the mistake myself of having specimens at my house, which is not air-conditioned, in Maryland in summer, and going back after ignoring them for a while and finding just rows and rows of fuzz where the specimens are, but I can't see them any longer. So that's a good entrance way into it. You do ultimately, if you're collecting specimens, it should be to give to someone who can identify them, or you'll pretty much need to get a microscope of some kind to get into the detailed ID. So this is not for everybody. It's really what I'm talking about is collecting specimens that can be used for identification. We do identifications ourselves. Usually other people who are getting involved just we prefer that people don't pin their own specimens. So we prefer that they send us packages of wet specimens that we will then process because we have our standards, I guess. But on the individual basis, what I just went through in terms of a net, soapy vial, canning jar, blow dry will get you great specimens, particularly if you process those specimens within the first 24 hours. Let me just say that bees will stop moving in soapy water within a minute. We actually have timed this. But they will also wake back up if they have not been in there for, let's say, several hours. So usually the best thing is to collect them in the soapy water, process them first thing the next day, if you have time. If you don't, you drain them and freeze them. So if, we don't bother with alcohol. So let's say I have a vial, which are centrifuge tubes of specimens in soapy water. I'll just go to the sink and I'll put my fingers as a block and let the water drain out. I'll put in a paper label in pencil and close the lid and throw it into the freezer. And then whenever we have time, we'll process them.
Sam, I'm just going to jump in because it's a little bit after the hour, and thank you so much. I mean, we're not leaving with Sam, but I want to thank Sam. And folks, if you have to hop off, I understand that. If you want to put a thank you in for Sam in the chat box, I know he'd appreciate that. And then we'll turn our attention to some of those questions that you put in the chat box, because there are a lot of good questions still in there.
I mean, there's so many things to cover if you want to get into details. And people can visit our lab too, I'll just mention as an aside.
There were a couple questions about genetic identification of bees and kind of where we are with that.
Okay, well, the short answer is that's a promising field, but we're not at the level in which I can turn over, here's a set of frozen bees, we can throw them into some kind of machine and come out with the answers of what they are and how many. That's probably where we will be. So we can then collect a lot of specimens, and a robot essentially would ID things. There's a lot of technical problems right now. So I would say we're still, if when we're looking at collecting and determining where species are and how many there are, we're still all old school, just what I was talking about. Pin specimens and looking at them under a microscope. We use, though, and send to the folks who do these kinds of things specimens all the time, so that they can run their molecular analysis and get a valid, molecular ID string, or whatever they call it, to put into their database. But we're not ready for to do that as a primary tool.
Okay. Cynthia wonders how much Dawn soap you use in the capture cups.
Oh, in the capture cup, just a drop is fine. It's pretty concentrated. We don't ever measure it. When we're doing out in the field, we show this. These are our favorite kind of containers for doing water. We have to add these weird labels to them, but that say, this is soapy water, don't worry. But ice, what is it? Arizona iced tea, some of the orange juice ones. You just want to not use milk jugs because they inevitably leak. And this nice handle here, this is great for pouring. So doing your bee bowls, that's your archetype of bowl, of water holder.
Are your preservation methods for photography the same as the preservation methods for genetic research?
Pretty much. So we are pulling specimens for our photography from lots of different places. Sometimes they're just so rare, we're just going to take a picture of it, no matter how ugly it is. But a lot of times we're like, oh, what's the prettiest one of these bees that we collected for that specimen? And they'll end up getting their photographs taken. I can, whenever we want, if we want to, at the end, I'll walk people over to the photography thing. I know that
there were questions about a few questions about that. Mary has a question about volunteers in your lab, and you have a lot of volunteers. Is that right, Sam?
I would say, yeah. I mean, we take volunteers for a day. Because of the kind of work we do, we'll put anyone to work. But yeah, always glad to have people. We have just lots and lots of things to do. I should also mention that we're basically also, in addition to the specimen-based things, we're also a plant nursery. So we're raising a lot of native plants so that we can plant them here at the Bee Lab. We now have 30 acres to play with, and we will start doing more and more intense surveys of individual plant groups, so we really can measure what it is that an individual plant species is supporting in the bee world, which you would think we would know, but we have hints, but we really have very little quantitative data.
Can you describe again why you use and how you use the rhinestones? There were a couple people who were a little unsure about that.
So the rhinestones are good because they can go through a wash-dry cycle without deteriorating. They're basically indestructible. And so each Petri dish, we have a whole series of Petri dish with unique combinations of rhinestones. What can't go through that wash-dry cycle is paper, right? So if we put the paper in, we just get pulp. And so we pull the label out, but and then we're running a whole bunch of different batches of specimens. But in the end, we have to know, like, that label goes with that batch. So the rhinestones are the connection between the two. And we glue, this is based on years of bad experiences. So we found that we have to glue the rhinestones to the bottom of a Petri dish so that things don't get mixed up.
Gotcha.
Does that make sense?
Mm-hmm. There were several people who were enthusiastic about seeing your photos set up. So do you want to do that?
Yeah. Okay, let's see. I think, oh, wait a minute. I just have to, I think I'm on our network. Let me go to Wi-Fi here first. I think it will automatically shift. We just have this, I want to do that. It says battery fully charged. There we go.
Oh, and somebody wondered where you are. I mean,
we are on Wi-Fi. Good. I was smart enough not to connect in. Where are we?
yes.
We are at the Patuxent Research Refuge. Oops. And this is the bee lab. So we're at Patuxent Research Refuge in the old endangered species area. This was a garage. The garage door is over there, replaced now with our beautiful mosaic, concrete floors, and we're sort of do-it-yourselfers. Who wants to? You should have seen this place before. It was just a nightmare in terms of an aesthetic nightmare. How about that? So that's Patuxent Research Refuge is between Baltimore and Washington, right off of the Baltimore-Washington Parkway, if that makes sense. It's a big green spot. It's lovely to work here. So I'm closing off the curtains, and I want to make sure that we're not blinded. So here's our setup. So what you're looking at here is a camera. This is just an off-the-shelf Canon Mark III, which is way out of date, but we don't need a lot of the features of that camera. We basically just need a large sensor area on there. There's a repurposed jack stand here, which is used in chemistry, just allows us to raise and lower this. There's a block of plasticine clay covered in black velvet, much blacker than black felt, as another little tip. And the specimen would go on here either to show the back or upright. And the camera is on a sled. Let's see if I can get a little closer here. You can hear the freezer in a loud way there, so hopefully you can hear it. So the sled is here. We have, because the flash uses, we're asking the flash, a lot of the flash, we have a power unit. I think it's the only one made. That's the quantum one that will run off of electricity. So you can buy battery, big batteries for that too. But this runs straight off of juice, and that will run the flash. There's a microprocessor here, which is not on, that allows us to program the sled. The sled will basically say, start here, end here, take a picture every so many microns. It runs through that, and then we take that collection of shots and run it through a software package. Over here, you can see some of the pictures that we're in process of there now. And it takes all those, sorry for the noisy freezer, takes all those pictures and recombines them into one completely in focus. So it's about focus. The lenses that we use, so this is a Canon 65 millimeter 1 to 5x. So it can go to 5 times the magnification of a normal subject. And that's great, except for the amount of, so in practical terms, like a couple of antennal segments are in focus when you take one picture. So it's not useful as a single shot thing, but if you take a whole series, then you can combine them, and that's what we're doing. And then there's a lot of Photoshop futzing around. We have some links and things for people if they want more information. This is not complete unless the beer cooler is on top of everything to add reflective surface. So there's a black velvet backdrop because we want black in the background. Let me just, too many things going on here. We would set this up if we were in action, but just to show you, we play around with different, what we call bridges over the specimens that are where we'll do different kinds of velum and reflective surfaces. And then ultimately we drop, I'm not going to do this well, we drop the box over the top, and the flash is bouncing around in the beer cooler. And wow, I'm not very good at this, am I? And we'll put to blacken it even further, we'll drop a velvet cloth over everything. And then we can hit the start of the microprocessor, and it will run through and take whatever number of pictures meet our criteria.
And there's a question, why black instead of white background?
It's a preference thing, but it has practical, I'm going to move away from the noise there. It has a practical outcome because that's a bad shot. With white, light fall off is an exponential function. And so inevitably when you're using white, you're not getting pure white, you're getting a whole series of whites to grays, even though it looks very white, and it's very difficult to modify the background then. With black, through some basic tricks, we can push it to absolute black. So most of our shots are going to be 000 on the color pixel chart. Sometimes we get that from being good at our setup and the camera is happy about it. But a lot of times, we just do that in Photoshop. And then we can place that specimen in any kind of black space that we want without having to come up with some connived border shading Photoshop thing that would be horrible. So that's why.
Okay. Bob asked if that is a stack shot. Yeah. Okay.
Yep. I don't know that there's other, so there's a forum called macrophotography.net where like everyone who is deep into this puts posts, and it's like basically nerdy white guys, and they're machining their own equipment. There's lots of different ways to do it, but for off the shelf kinds of things, I think stack shot is about the only game in town. I could be wrong now.
There are a variety of people who would like to give your lab money. And so they would like to know how to do that. Is the Discover Life Plistes link that Elizabeth put in, is that the...
Yeah, Plistes Foundation is where money can be donated. That's great because I am actually the only employee here by the federal government, other than Claire Maffey, who works for Fish and Wildlife Service, who just joined us. But our technicians, our lab manager is paid by, soft money or out of my pocket. So that would be awesome. And I'll just say, expansion wise, we're expanding to lots of outside plant things. We have a good volunteer network of just the plant people. And there is talk of a national pollinator center getting placed here on the refuge as a possible, multiple year new kind of initiative by the Biden administration. We'll see how that goes. But oh my gosh, so much more interest now than in the last 4 years about bees. So it's good. A little
Eileen, sorry. Eileen had a question about your work on power line corridors, and she wonders if you have any updates on how U.S. energy companies are moving along with that, particularly with monarch habitat.
Yeah, so monarch habitat, I think, is probably actually more of a game than because it's just got more, let's call it public cachet. It's something that people get like right off the bat. Oh yeah, of course, monarchs. Whereas native bees, people are ramping up to that, but they also don't have a lot of like inherent understanding of how that works. So a lot of the monarch thing indirectly is going to benefit most of our plant work. But what we'll want from power companies, and here at the refuge, we see a lot of rare and uncommon bees on managed power lines that don't just herbicide everything out. So the point is that these power line corridors are super important as scrubby habitat that in a matrix of either forest or agricultural, industrial, man-made things. And so we have, if you do Jared Fowler and Droege, you'll find it. It's basically Jared Fowler's work, 2 R's and Jared. And there's lists of specialist bees. So these are the kinds of plants we're looking at more intensely to act as, so that the biodiversity of bees is supported by the biodiversity of plants.
Trying to get that link real quick to put in the chat box.
It's done for the whole, Jared has done it for the whole country now. So all of North America, at least all the US, has at least a start of a list of bees that are super specialized. And by that, I mean like willow has 8 species in Maryland of bees that only go to willow. Now it might go to several different willow species, but if you don't have willow, those bees are gone. We can look at cacti, we can look at prickly pear. Dodder, of all things, has at least 1 species of bee that only goes to dodder. So more and more we see biodiversity as a general strategy for conservation as being important for bees too. It's not just plant clover and job well done.
Sorry, somehow I got the wrong link for the specialist bees. Give me just a sec here. So Sam, what I wanted to, okay, monitoring lab, that's still not doing it. Sorry guys, my copy and paste is not working. Maybe somebody else can put that specialist bee list in there. I keep getting your Flickr page. It's over.
Yeah, if you do Fowler and Droege. Droege is just an uncommon name. So
there we go. Claire put it in. Thanks, Claire.
There we go.
So there's a source with the bees, and then there's a source with the plants. I use them all the time. Yeah. It's really great.
And this is where we're going. We'll be providing more and more detailed lists of like the value of different kinds of plants in terms of, and the bees they support. It's a year's effort. There's a reason that these lists, this detailed information doesn't exist, because it's time consuming. You want to replicate it in a bunch of places. We will have a bumblebee survey, floral survey that keep that for people to look at. We've developed a technique now. I think, Denise, you're maybe aware of this already. And next year, we'll be releasing that to general use and have a little program, at least for the Northeast, for looking at how bumblebees divide up the world, because even understanding that that's a bumblebee and that's a carpenter bee, but I don't know which bumblebee, is going to give us some pretty important information. And it's the kind of thing that can be done anywhere. And your garden is one of the better places. You have a set of plants, bumblebees make choices. What's their choice today? Little half-hour walk around the garden is really all we're talking.
And Sam, I'm going to pull you back in then next year, because we're going to do a monthly Bombas bee course, a bumblebee bee course.
Oh, yeah, definitely.
We'd love to have you.
I'd prefer, if possible, just to get people throughout the year as to, do that earlier rather than later.
Mm-hmm. Great. So I'm going to end our session. Sam, I want to thank you so much for taking us around the lab, all the information that you shared, not just this morning, but in all that you do. You're really a great example of how science can, produce things that can be really used by a lot of different people. I'm going to share this screen. So this is the screen, some of you were asking about the site where you can make a donation if you'd like. And I wanted to make sure this was in the recording, since the chat isn't easily accessible in the recording. So
yeah, that's great.
Put that up there, and
extra bonus for giving this talk.
Great. So thanks again, Sam. Everybody, thanks for coming, and we'll see you next month.
Okay. Thanks, Denise.
Hi. Thanks, everybody.
Bye, everyone.